All right, well, we can see the bottom.
Oh, nice. Bottom in sight. Looks sandy from up here.
Alan and Stephanie, are you there?
Yeah, we're here.
I don't know if you've seen that question from John Reed in the chat, but he's wondering what sonar screening he's looking at.
Yeah, I'm checking with Roland right now, but he's a little busy, so I'm getting it in a second. Whether or not the sonar on feed 3 is Sirius or the ROV.
So the sonar that you see on the lower right quadrant, the blue view that you see on the left is on the ROV, and that's what we refer to as our multi-beam on the ROV. And on the right, what you're seeing is one on the ROV, and the right one is on Sirius. So the half circle on the right is the ROV, the full circle is Sirius sonar.
Thanks, Roland. Looks like we have a little eel here at the bottom. We'll get there soon. What, 14, 18 meters right now?
goes something. Like a little Medusa floated by there.
Like a little shrimp or something in front of us. Maybe a fish. He doesn't have a lot of control, though. Floating by. The sand looks kind of wavy from up here. Oop, something red.
Mm-hmm. Future collection.
Maybe. I don't know
See
what it is.
that animal may be captured in our environmental DNA samples.
Yeah, maybe. Looks like a fish there floating by.
So as a reminder, we'll be taking environmental DNA collections, basically just a water collection at the beginning, middle, and end of this benthic transect.
And then again at 500 meters on our way up.
Yes.
There's a critter. Maybe he has some hard bottom here. Go ahead, Nev. Okay. Is there a possibility we'll end dive early, or is it not that serious? Okay.
Hi everyone, this is Matt,
Thanks.
science lead, expedition coordinator on the Okeanos Explorer. If you're tuning in, we are having some technical difficulties with the ROV right now. We're hovering above bottom and troubleshooting to see if we can continue with the dive. And as our expert ROV team looks into that, we will just hang out here and hopefully we can continue to dive, but we will keep you updated. Thank you.
Thanks, Matt. Maybe we should just do a still midwater.
Oh, yeah.
Sit here and look at stuff. There might be some hard bottom over there. I wonder what that is. That's promising.
Yeah, over to the left, yeah.
Yeah. Rocks or something. If this was a Lophelia mound, I would think there'd be Lophelia down here, but the head tumbled off. But it's still too early to tell.
Yeah, I'm not exactly sure how close we are to the mound right now. I feel confident this is going to work out.
Yeah, I hope so.
Feeling optimistic.
Good. That's all you got out here is optimism. Hope. Oh, look, rocks.
Yeah.
Yay, rocks. I know. We might have to do a rock sample.
That would be a good idea.
Maybe look for a small one. Looks like there's some. Yeah. Looks like we're getting close to the bottom here. You can see the slope going up.
Yeah, up to the right.
Yeah, up to the right. Which means we're probably facing. Okay, we're facing almost south. Does that make sense?
Yes.
Jelly, I see a jelly. I think it's a ctenophore. I saw combs.
Little combs in there.
Sparkly combs. A jelly of some kind.
It was a jelly. I was checking out the chat room. Silly me.
Here's something. Pyrosound, maybe? Nope. Ctenophore?
I think that is, lobate ctenophore.
Sweet.
Probably Bathycyroe.
I think I see a sponge.
There goes another Bathycyroe. Oh yeah, I see that sponge out there.
That was a cool fish at 6 o'clock. What do you think this is at 3? It looks almost like line, but think that's human debris over there?
Difficult to tell, but yeah. I can
We're
see
pretty
what you're
here too.
Is there more of it straight ahead?
Yeah. We're pretty far offshore.
I'm going to guess that it's sponge stalks.
Could be sponge stalks.
Of course, that's an engineering video.
Roland, I thought they were like a couple feet long.
Oh.
Seen some that have been about 3 feet long.
Oh, neat.
So it'd be nice to get some of the rock probably from the bottom and the top of this structure, I would think.
Hey, did someone call in?
Hi, Stephanie, Scott, friends calling back in. Glad to see we're back on the bottom of the ocean.
Yes, too.
It's exciting.
There could be some good rocks to grab here. Yeah, these look like slabs, right? So I'm not a geologist, so you're going to get a very elementary version of the habitat out of me. But these slabs are pretty large. They're like 1 to 2 meters wide, maybe, and they're fairly thin. So more like of a pavement, like a chunk of pavement, than necessarily like a rounded boulder. We got a nice white coral at 3 o'clock. Looks like it could be a gorgonian.
Yeah.
So the sand looks like it's fairly thick underneath these rock boulders. Right, like you could sink your hand into it. Really,
your hand through it.
Yeah, it's probably pretty soft.
We may see when we make a rock collection.
Yeah. Which is similar. That this is that thick sand we saw on the dive yesterday when we hit bottom.
Yeah,
thick
nearby
sand.
here.
Yeah. There's also some yellow, I believe, gorgonians or nidarians at 3 o'clock on these rock tops.
Seeing all these rock slabs is interesting, and it makes me wonder if we are going to be wrong about this being a giant coral mound, or if it's some sort of interesting geology out here.
Mm-hmm. My guess is this isn't a coral mound, just because I don't see any Lophelia rubble. And usually at the base of all these Lophelia mounds is always rubble that has, rolled down or whatever. The Lophelia mound's actually built out of that rubble mass. So I'd be very surprised if we have an actual bioharm Lophelia reef. I'd be more apt to say this is probably some kind of rock feature.
Yep, I agree, and we saw that on the first dive, didn't we, when we settled down below that
yes, please.
That bioharm.
Mm-hmm. I think I see Stichopathes at 9 o'clock. Okay, that's water sample number 2. Did that one take?
I gotcha. Thanks.
It looks like maybe a yellow sponge up here. It could be a yellow hexactinellid.
The Hertwiggia, maybe?
Possibly. And then I also have a small coral at 3 o'clock, little white fan. Looks like it could have an ophiuroid on it. And then in the distance, okay, yeah, so that's at 3 o'clock. So if you swing right,
think.
Number 3 fired.
Number 3 fired, yes, and number 2 it just stayed open.
Number 2 did not work, so we are... Okay. So that means we only have 3 samples left.
2 samples.
2 samples left.
Middle and end of the benthic transection.
Right, because we did a 500 and then fired 2 but didn't work. And then number 3 is really this bottom sample.
Yes.
Then we have 2 left. Okay.
Great. Now we get to look around.
Yeah, if you guys are ready to start looking. Okay. Yeah, if you guys are ready to start looking. Okay. Yeah, if you guys are ready to start looking.
Yeah. Fantastic. Fantastic. Fantastic.
It might be someone shoreside with their phone on. So again.
Possibly.
If you're shoreside and have called in, please mute your phones. Thank you. Yeah, there's a Coral dead center right in between your lasers. If you could just zoom in on that guy.
Oh, we've got the lasers on, so we have a better feel for the size of these slabs. This one's definitely more than a meter. And we're going to get a look at our first benthic organism here. So we have a nice...
Is that bamboo coral?
Bamboo coral, maybe?
And I would call that astroschemia, but I'm sure... oh, and a Faria, look.
Yeah, you see the glass sponge, Faria Octa, probably.
This is looking like a Primnoidae
Primnoid.
Octa Coral to me, not a bamboo coral.
Okay.
A very common deep sea family. And I'm even seeing something wave behind it that is unbranched.
Oh, yeah, that yellow.
If that's, I don't see it yellow, but maybe you definitely have a better view than me. Yeah, that must be a Stichopathes said earlier. But yeah, this looks like a primnoid. You can see that the polyps are arising in these whorls. I can't tell right now if it's 2 or 3 polyps per whorl. They're sort of connected near their bases. Very classic.
It looks like
pattern.
3. I see 3 per
node?
Yeah.
Yeah.
Yeah, so not sure if this is genus Paracalyptophora or Calyptophora. Have to look into that a little bit more. I'm constantly forgetting my primnoid genera. There's a lot of them. That's great. We have a black coral and an Octa Coral next to one another.
Yeah.
Nice.
Yeah, it'd be great to take a look at that.
Sponge. And this is, my guess is astroschemia, but I'm not an expert in these. Long spaghetti star. It's a type of ophiuroid or brittle star. They're usually a sieve. If I am telling you any wrong information, I'm sure Chris Ma will chime in and yell at me. So I will be corrected shortly.
No worries. I think there was a whip behind this as well. That would be worth taking a look at.
If you guys can look up at around 2 o'clock, there is a nice white, wrinkly sponge. Yep, top corner. There might be a shrimp here underneath that Stichopathes. Yep. Ooh, look, maybe a corallimorph or a solitary coral here.
Right on the top of that rock
Yeah,
to the right.
At 3. Squat lobster. Oh yeah, squat lobster. Look at that.
Yeah. Oh, we got lots to look at.
We got plenty of time, so.
As usual.
This is a short distance dive. Like, we don't have a lot of area to cover, so we can, chill and zoom in a bunch of stuff.
Zooming in on this glass sponge, this is almost certainly the genus Pharea. And what's that just to the left there? Is that a small urchin?
Yeah, it looks like it to me.
I like the looks of that urchin.
Yeah, he's cool.
John Reed is commenting in the chat that it's possible that this is an allied group, Eurydidae.
It's thicker.
But it is a bit thicker than a lot of the Phareas. I think he might be right on that. Be good. Can we zoom in on that urchin a little bit? I think we'd like to get some imagery of that.
There's actually quite a bit of stuff on this rock when we zoom in.
Sorry if that was max zoom.
Yeah, he's tiny.
Oh.
He's a tiny little thing. There's also a white-tipped, I would call this a solitary coral, is my guess, on the left. I'm sorry, the right at 3 o'clock. But I can't really see its helices, so I'm not really sure.
Rather than a corallimorph?
Yeah,
maybe?
I think so, but I'm not 100% on that. I guess I'm not 100% on anything I see down here.
I understand. Same. But so corallimorphs are a lot like, they're the sister group of the scleractinian corals, and their main difference is that they don't have a calcium carbonate skeleton that they're. So we should be able to tell.
That's cool.
That is really cool. Now, is there a skeleton in there?
I don't see one, but it's like a clear anemone. Bubble tips.
It's quite.
It's very squishy.
It's squishy. It's beautiful. But I'm curious what you can see the color inside. Is that it's not a red pigmented gut, is it?
It looks like it here.
Yeah.
It looks like this ring on the outside, though, is probably not calcium carbonate. That's probably the rock behind it.
Right. So this probably is a corallimorph and not a solitary coral.
It looks like a green sponge here at 9.
Yeah, you can see the oscules, the excurrent openings of that green sponge just to the left of this beautiful corallimorph. It's fantastic. And there's another sponge just to the top left of the green sponge as well. Another demo sponge, a white one.
Did we do an on-bottom Niskin? Yes.
Yeah, we did that. Yeah. So our next collection will be our third.
Looks like there's some cyanobacteria, or there was a squat lobster, but I think he's disappeared. Yeah.
Now the small yellow Octa Coral, is that the Gorgonian and
at 9?
Maybe Paramuricea or something like that?
It could be.
We could take a look.
Yep, he's flopping around in the breeze right there. He's right at noon now.
Yeah,
Right above laser.
Yeah, that's a black coral.
Oh, that's a black coral.
Probably
Thanks.
the genus Storapathes. I think you'll see when you get closer, the polyps look quite a bit different from.
Yes, they will.
What genus did you say? Oh, yeah.
Storapathes. We've already entered it into the annotation
thank you.
and the chat room.
Thank you. Oh, nice. So black corals are really Octa Corals. They have 8 tentacles, but they have a
No, they're not.
They're not
No,
Octa
they're
Corals.
hexacorals.
Hexacorals.
Yeah, they have
Hexacorals.
six
One of
tentacles.
the cool things, actually, this is a great view, actually. Each polyp has 6 tentacles, and what you can see is the polyp is all stretched out along a branch. It looks like the tentacles are coming out in pairs, and where you see that little red tube sticking up in the center, that's the mouth leading into that weird stretched out polyp. So it almost looks like polyps with 2 tentacles, but they're actually 6 stretched out.
6 tentacles.
Yeah, that is a great view of that. A lot of times it's a lot less clear.
In fact, I always tell my students it's really bizarre. I believe black corals are the only hexacoral where the polyp actually has 6 tentacles. All the others, there are always multiples of that.
Mm-hmm.
So true hexacorals.
Mm-hmm. Then in front
And
of
then
it is
the whip is another.
Yeah, likely Sticopathes. The black corals, yeah. It's yellow. I don't think it's Leucini. Leucini is the one we have in the I would say shallow water, but mesophotic. This one's yellow.
Yeah, and you can see how that's a little harder to see the polyps with 6 tentacles.
Looks like 2.
Mouths are more difficult to make out because the polyp sizes are so much smaller.
What do you got there? A white thing on the rock. There it is.
That's it's an octocoral as well.
Okay.
You can see the
Polyp.
just to 9 o'clock.
Barnacle?
Where are you looking? What?
It's got too long a stalk for a barnacle. It must be a recruit.
Oh, at 9 off. Now it's off screen. It looks kind of like a Gorgonian, maybe.
Oh.
Yeah, the little white one that we were talking about.
Maybe like olive fruit or something.
Yeah, it looks like it could be a recruit. There we go.
Gonna zoom in there.
Yes.
Oh, I see. It's actually got 3 or 4 polyps, doesn't it?
Yeah.
Yep.
4. 1, 2, 3.
Well, that's a good sign. It's telling us that there's recruitment here. It's
Yeah.
not just all old corals from long ago.
Mm-hmm. It looks like there's a little brown guy back there, maybe a clam or something. Okay, we're happy whenever you guys want to move on. We're taking our grand old time, though, because we've got a short transect today. So, lots of zooming and stuff.
Oh,
And I think we'd like to collect a rock, so if you find one that you guys feel like you could pick up real easy at the claw and will work for you, we're happy to take really any one you want. I don't think so. Just
Hey, Stephanie.
Yeah. Hey, John, what's up?
When you're looking at the rock close up, it looks like the typical deep water phosphorated coated limestone.
Yeah.
And it's pretty common out here. I don't know if there's any geologists on board, but
Not on board,
like
but
that
maybe on the
phosphorus
chat.
coated.
haven't seen someone in the chat necessarily yet.
Yeah, so like the
Like
Miami
a-
Terrace is made of this block, like plaque, I guess it's phosphate, phosphoritic, right? So this, it's the common rocks. This is interesting though. There's like a shale shape.
Mm-hmm. Looks like more like consolidated of sedimentary form. So So I think we're lining up to do a rock collection.
Mm-hmm.
It's too bad that corallimorph didn't look easier to pick up. Scott was commenting that he thinks that of all the cnidarian groups, they're the least well collected in the deep.
Yeah, the little bubble tips.
In fact, Alan, I can't think of a single collection we've made of a corallimorph.
Well, we can go back and get it, or try to get it.
Well, I think it needs to be on a substrate that's easily picked up. Otherwise, you're going to have to scrape and claw, and who knows what kind
Right.
of shape it'll come in.
So if we find one on a small rock, it might be worth it.
Yeah.
Exactly.
Yeah. This one looks like it might have a little lollipop sponge on it.
Oh, a little clatorizid, maybe?
Maybe. And those are the carnivorous
Yes,
sponges.
those would be carnivorous sponges. So unlike most sponges, well, let's see when we get a little zoom in here. But if it is a carnivorous sponge, that would be pretty exciting.
This is going to be sample number 3.
$3G, yeah?
All right, so the sediment's really fine. You can see it just clouds up when we touch it. That works for us.
Nice. Well,
Some sponges on it.
definitely some sponges. Little ball sponge. Reminiscent of Geoda Day, but
Oh, geez. I can't.
who knows?
Yeah.
Right?
Well, we'll find out when we bring it up. A good underscope
And
if you want.
I can't tell, those look like maybe dead hydroids blowing in the breeze there. They've definitely got a lot of sediment collected on them. On the other hand, it's possible they could be alive, and we will examine those when we get them back to the ship and preserve them if they look like they're alive.
Maybe tiny little corals on this rock. Some solitaries or gorgonians or something. All right, so it's about a 10 centimeter rock.
That's going to be great to look at.
It's like a cobble piece. Cobble size. There's lots of stuff growing on it.
Yeah, it looks like solitary coral there. Dead center, right?
Yeah, it could be. Some neat little sponges.
Yeah, got a lot of little things growing on there. Lots of sponges.
Maybe some worm
I
tubes.
think you guys are going to be up late tonight because there's an awful lot on there. I think a black coral as well, maybe.
I told Mr. Collect It over here that he's not allowed to fill every bucket. So.
Well, we will.
Alan, I disagree with.
You agree with me?
Agree with you. But I'm just saying, yeah, this rock alone has got all kinds of wonderful stuff on it.
Mm-hmm.
Yeah.
No, go for it. Rock box is good. You doing port side?
Looks like a success. No, that's the first rock sample. Well done. Great collection.
We have a pretty cool fan at 3, too. I'm not sure if that's a coral or a sponge. If you guys want to move there when you're done, when your arm is stowed and you're comfortable to get moving. That looks like Stickopathes to me in the center. This orange whip coral. I'd have to get a better look, zoom in, and check its polyps out, because we also have like an Eli Zelid that looks similar to that. It's whip-like.
Yeah, I agree with you, Stephanie. That's going to be a Stickopathes.
Yeah.
Something in the genus Stickopathes or closely related to it, black coral.
But this is separate from the other whip that we looked at.
Yeah, I think the other one was a yellowy color, much smaller. We get them in gray up in shallower water. Go ahead.
Yeah, there are currently, I believe, 2 whip genera, Stickopathes and Cirripathes, but some of the genetic work has shown that there are actually 2 pretty distantly related Stickopathes groups. And so I think this orange one belongs to the other one that may be closer to the family Aphanopathidae. I don't remember exactly, but it's still called Stickopathes. But for sure, we have 2 different species right here that we're looking at, the yellow one.
The yellow and orange, yeah.
We have that same problem with hydroids that
oh, geez, have
don't
it with
branch.
everything. Have it with everything. Yeah, the white fan at 3.
There goes
it's just right at the edge of the screen there.
Bathysiroe floating by.
Nice.
Looking like food to some of these filter feeders, maybe.
Just from a distance, my guess this is going to be a type of Phycalia or elephant ear sponge, but we'd have to get closer for me to really confirm that.
Fish in the top left there, eel.
Yeah. There's also something brown here and coming into the center screen around 4.
Just below this fan.
Not sure what that is.
Yeah, when you look close at these rocks, there's a lot on them. Gonna come in for zoom here. Nice.
So if we look on the other side of this, it might have that veining.
The elephant ear vein.
Yeah, I don't know if you'll be able to get to look at the other side. It might be in an awkward place for you guys, but yeah, only if you want, can manage. Otherwise, if it doesn't have that veining, I'd probably call it something similar to Pacastrelid. If it does have the veining, I'd call it Ficalia.
Maybe Christina will call in.
Yeah, I don't know if she's watching
She
today.
recognizes this.
Oh, it looks like garbage down there, too, huh?
Plastic.
It's very thin, that fan. Yeah, that plastic bag is awful.
That sponge.
This is Stephanie.
Yeah, John.
Christine is on a Zoom until about quarter after, I believe. So we'll give my expertise of spongeology. When you zoomed in on it, it looked very, very porous, like a hexactinellid. But from a distance, it did look like a Pacastrelid, that Pacastrelid.
Yeah, and we're looking at the opposite side of it now, John, and I don't really see that veining. Usually that veining is really like a vein, like really distinct veining.
Yeah,
So you're thinking Pacastrelid, John?
No, he was wondering if it might be a glass sponge.
A glass sponge.
Yeah, this view, it looks like Pacastrelidae.
Okay.
The other side, it looked very, very porous, like a hex. So
Okay.
How it's kind of fringed around the edge, I'll vote on Pacastrelidae.
And there's that stupid plastic bag.
Yeah.
Yeah, we're going to call this Placastrella.
Maybe. Okay, I think we can
Yeah, we're
pull
good whenever
back
you
and look
guys
at
are ready.
something else. Now, do you want to see the brown organism that was a little bit closer to the ROV from here?
Yeah, if you want to, I'm happy to do that.
Sort of bushy about one rock back. Let's see, maybe to the left of here. Or we can keep moving our way up. I'm sure we'll come across, well, shouldn't be sure that we'll come across that exact species again, but we can hope for it.
There's also this blue. You were saying you thought that was Mycali?
No, I think, I don't know, but there are shallow water blue sponges in the genus Halasarca.
Halasarca. We have another Gorgonian. It looks like at noon. It's been flopped over.
Yep, more Stichopathes.
Mm-hmm.
And the yellow, that was Stauropathes, was that correct? The bush-like black coral? We just went out of view.
Yeah, that's right, Alan. Stauropathes, kind of like a little fan.
Yeah, thanks. I think that, yeah. Wait a second.
Not this one.
No,
This
that's
is a
not. That's an
Or at least in
octocoral.
the family Paramyricidae.
Paramyrcid.
That's no longer. Yeah.
Great.
It's actually now the family Plexauridae, but something like Paramyricia for sure.
Mm-hmm.
So this is an Octa Coral, this one.
So that's new.
that white thing on top of the rock there looks almost like a stylaster knocked over.
Yeah, it does, doesn't it? And I also think I see a stalk crinoid at 9 on the rock that's right below us. There's also a fish down there. You can see his black tail.
Can you zoom in on that coral that we just passed on the right?
Yeah, we can come back to it. Since we just blew past it, I will come back to it, John. Yeah, can we look at this? It's at 1 o'clock.
It's really nice stock coral.
Crinoid.
Yes. Did I say stock coral?
Yeah.
Stock crinoid. Looks like it has an associate on its stalk there.
Yeah. Oh, neat. So this is a stock crinoid.
What is going on there?
Looks like a hermit.
Is that a hermit?
Mm-hmm.
Oh, I see. He's holding on. It's got a shell.
That's a great view. Hey, before you pick up and move away from this after you've got this, just look off to the left on the next rock. There's, I don't know if it's another one of these stock crinoids, but there's something odd. I almost thought it was like a Metallogorgia or Chrysogorgia colony, but
okay.
Too far away to tell. Thanks.
Yep. Cool.
You don't usually see the hermit crabs up on these crinoids. This is a bit of an unusual observation.
Mm-hmm.
Now, as we go back and get a better view of these rocks, we've got a comment on the geology here that these look like sedimentary rocks which have been exposed along the continental slope, and the black color is ferromanganese crust created by minerals that are deposited from the seawater on the surface of the sedimentary rock.
Awesome.
The crust grows extremely slowly, so these rocks have been exposed for quite a long time.
Hey, pilots, can you go back to that flattened stylaster white fan coral that was on the rock before? It should be to your right.
Hey, wait. Before you go, Stephanie, all you have to do is zoom just to the left of where you were looking.
Zoom to the left. Okay, wait.
You don't have to come
before
back.
you run off. There is a 9 o'clock coral real quick in this shot. Sorry, I'm getting bossy, guys. Okay. I get to get really pushy back here. It's real easy to tell them what to do. Yeah, 9 o'clock, it's a stalk on the rock, right next to the rock we were just looking at. So it's just about to come into center. Yeah. And what do you think this is, Scott?
Well, I wasn't sure. It could have been a Metallogorgia or Chrysogorgia skeleton or just another whip with hermit crabs or something on it. That's why I wanted to have a look.
Yep, dead center. There you go.
I was hoping we'd move that we could just zoom.
It looks to me like a dead thing with a Hermie.
Okay, so it's the same sort of thing. We've got a hermit crab again crawling up a stalk.
Mm-hmm.
Maybe it was a crinoid stalk, but this is interesting seeing two of these next to each other. I haven't seen that before, and then all kinds of hydroids.
Oh, it's
All right,
so many
great.
hydroids.
Mm-hmm.
I like it.
Okay, I think we're good, pilots. If you can go full wide, and then there's a toppled over stylaster we want to get a quick look at to the right. It looks like a white lace coral. Yeah, ahead there's something crazy going on too. Yeah, there's that one's good. That one that's standing up. But John was in particularly looking at the one that was toppled. It looks like another Faria there, maybe.
Yeah, another glass sponge. If it's a euretid, might be.
He should be on this rock right here, I think. Or near it. There it is, right coming up into screen, yeah. Ooh, I see a sea star. Right there.
All right.
Yeah, that's good if you could snap zoom for us.
Calling Dr. Ma.
That's a pretty big one if it's Starlaster. I think it's Starlaster.
It has that.
Oh, no, it looks like it might
No?
have polyps. Yeah, it looks like a toppled gorgonian.
Yep.
So that's interesting. Still alive.
Little shrimp hiding under
Yeah,
it,
a little something behind there.
I think.
Shrimp
Yeah,
or
this looks
fish.
to me more like a corallium or hemicorallium, something like that.
Corallium.
So definitely an Octa Coral. One of the precious Octa Corals.
There's a little hermit under there as well, tucked away. Now, where was that star you saw?
The star is below it, like more towards the 6 or 5 o'clock time when we're zoomed full wide.
And I can confirm that Chris Ma is in the chat room.
Okay.
And if
Here's
he's,
another shrimpy.
yeah.
Yeah, there's a star, a little white star. There he is. He's just
Very thin
dead
arms.
right of laser. There's a neat, looks probably a hexactinellid below.
Oh, gosh. No,
Really thin. I just
was-
call those ice spongage because they look sponges because they just look like a layer of like crusty ice to me. I don't know what they are. They're not they're not uncommon though. Those little thin crusted white hexactinellids.
was it sticking up or encrusting?
I think it's encrusted like
Yeah,
this,
that's-
like flat to the.
pretty unusual for glass sponge.
It might not be glass sponges. It might be a demo. This real
So
thin.
It's be interesting to look at this star. Chris comments that it's appears to be it's sort of hunched over, like it may be feeding.
Yeah. That's neat.
Can't see what it's feeding on.
Rock schmutz, I guess.
Maybe.
Nice. Did he have a name for it?
No name is forthcoming so far from the chat. But Henricia.
Oh, okay.
Sorry.
Yeah, the brownish discs. I don't know what those are.
Would be nice if we can. I'm not sure we can get any closer, right?
That's max zoom.
Yeah.
Yeah.
Yeah, so if this is Henricia, Chris reports that it's most likely eating a sponge.
Okay.
Of which there appear to be many around.
Yeah, I think that's probably what these little, like,
Yeah,
dime-sized things are.
Mm-hmm.
Little white sponges, little brown sponges.
Thank you, Chris.
All right, we're happy whenever you guys are. It looked like ahead of us there might be a really cool, like above, you'll have to pick your head up, but yeah, far in the distance there might be like a long stalk and like a fanny thing on the end of it, so. The fanny thing. Yeah, coral fanny thing. There's also maybe a cutthroat eel. This is interesting. Single stalk with like a top with like a bush. I'm not sure what it is.
Yeah, I feel like I've seen a picture of that somewhere, so hopefully Scott will chime in and let us know.
Almost looks like a hydroid on the end of a Sticopathes, but I don't have a clue. That's odd. There's another one behind it. It has to grow like this, whatever it is. Ooh, and way up there at noon. And the way back up top.
Big fan in there?
Yeah, if we get a zoom in on this guy and see if we can get an ID from shoreside. Oh, there's definitely a Gorgonian. Oh, weirdo-rama.
So great. Another one of those brittle stars in it.
Yep, sorry guys, I'm on a separate call as well. So this is Metallogorgia classic Octa Coral, and it's always found with one brittle star. We know from around the world where this has been collected that one species of brittle star lives only on this colony, on this species of coral, and the species of coral only ever has this one brittle star associated with it.
And do they always have one?
Association. And this is on the shallow end of the range for Metallogorgia melanotrichos, so it's kind of interesting to see it here. It's clearly doing really well.
Yeah. So I think there are two of those, right? We saw one or something that looks quite similar behind.
Mm-hmm.
Some of those polyps look distended.
This is a really nice close-up, and there's some kind of funky branch tips there. Those curled ones that you see on the right side, look like they're extensions of branches. That's kind of odd. You notice everywhere else it's got a very regular sort of bifurcating growth pattern. Gives it this very geometrical shape. So I'm not sure how to explain those wiggly curled ones.
And they're also lacking...
I've never seen that before.
Lacking polyps? Or are the polyps retracted?
I mean, they're covered in tissue. They're alive, and then, you can see some polyps at the very tips of them. That's weird. That's really weird.
That's what I like to hear.
Okay. Yeah, me too. Thanks for finding that.
Hey, Scott, can you type that into the chat?
I've got it.
Just so I can spell it.
Yeah, I think I already did.
You did? Okay. Thank you.
Right after my comment of, ooh.
Yeah. And then, there's another one behind there.
So this we think is maybe in the shallower end of the other depth range. Ooh, there's some, I guess, a cutthroat here. There's another one here.
Yeah, Stephanie, I don't know if I misheard you there. Normally they're deeper, so this is the shallow end of the range.
Yeah. So that's looking at its stem. It's very textured.
Yeah. It's an interesting bend in it. Oh, this is just...
There's some crazy stuff in here. Okay.
Cool.
I have my eyes focused on noon in the distance. It looks like a really tall fan. Okay. If we can make our way that direction, that would be cool. Ooh, hello, shrimp. I mean, those elbow shrimps we saw yesterday.
Today, yeah.
Those really sharp elbows in their antenna. Okay. Oh, nice.
Oh, let's look at that sponge. I want to collect it.
Of course you do.
This is a stalked glass sponge.
It's not Hylanema, though.
No, I don't think it's Hylanema.
Hylanema has the really long stalk, but they're like this broken up, like pieces of fiber optic cable, it almost looks like.
Right.
This one is more
stalk.
It's stuck to a rock.
Fantastic.
When it's got critters in it.
Oh, that would be great.
You want to get this guy?
Yeah, is that collectible? That would be terrific. think the stalk itself is going to be hard, but the top looks like it's going to be fairly
neat.
soft.
Okay, so we're going to be wearing gloves when we get this out of the, for
Yes.
sure, because those spicules are going to go right through your fingers. So to be very careful when we take this out of the bucket.
Oh, yeah.
Sometimes they go right through your gloves.
Right.
Do you have a name for it?
I am afraid that I do not.
Okay. Hexactinella is good?
Well, it could be one of these, Hexactinella. There's a couple different families that have stalks, even within the Euplectellidae, that some are stalked. But the taxonomy to figure these things out is in the details of the fine spicules that are in the organisms. And so
we're on dive 5, and this is going to be sample number 4.
Yeah, this is sample 4B.
I would direct them, let them
please.
know
Yeah.
if they need to get the stalk if that's what you want.
It would be good to grab it by the stalk if possible.
Okay. And then it's going to be probably pretty crunchy.
The stalk itself should be very hard. The top, I expect it to be
Delicate.
kind of soft. Yeah.
Okay.
Oh, it's bigger than I thought.
Yeah, so it's about 15 centimeters on the top. Yeah.
Yeah.
That is a huge oscule, too, that big hole in the top. That's the excurrent siphon where the water comes out.
Yeah, that's the excurrent.
Yeah.
Yep. So the animal is drawing water in through the sides of its body.
And that stalk looks like it's almost fleshy.
Yeah, it's made up of parallel giant single axis spicules all together. There are some that grow just a single giant spicule upon which, but that's not what we're looking at here.
This is
made up of multiple parallel spicules.
So if we were to cut it, you'd see all them kind of like fiber optic cable
Exactly.
growing in that, almost like a rubber. It looks almost like a rubber hose, right? I don't know how to explain it.
Well, they have this
soft on the outside and then real sharp spicules inside.
They're actually secreted intracellular, so they're inside the cells. The cells surround them, and they secrete it on a non-mineralized core, and so that creates a little small space, and then it's laid down on top of that layer after layer.
Neat.
So that's what gives it the property, fiber optic quality.
We thinking undescribed species or just a target interest?
I would be surprised if this is described, but,
Okay.
but a lot of the deep hexactinellid species that we know of, and there's 500 or 600 described ones, they have been described based on a single sample.
Oh, geez.
So they really are you can imagine, since they're found mostly in the deep sea, they're very undersampled.
Plus, and those samples probably came up on nets and net trawls, in which case, if this is coming up on a net trawl, I mean, it's getting destroyed.
Yeah.
This is like a fragile. Most hexactinellids are super fragile. So pulling that up on a net is just forget it.
There are exceptions to that, but yep.
Do you think they'll need to clip that?
I think it'll break off.
I think it'll break off. Okay.
But I'm new to this.
I mean, usually, if you've touched them in the lab or have handled them or something similar texture, you can kind of tell just from looking at them what they're gonna look like. Now, not always, right? Sometimes I think a sponge is gonna be rock hard. We use the manipulator and touch it as a rubbery thing, So
this is gonna be it's like cotton candy.
Cotton candy on a stick. Yep, that should work. Agreed.
It'll definitely be like cotton candy to some lucky researcher who gets to study this sample. This sample is going to go back to the Smithsonian National Museum of Natural History, and there it will be available for study by scientists around the world.
Mm-hmm. And we're going to take a DNA.
Yeah, we'll definitely take a
one?
DNA sample, and we will either sequence individual genetic markers out of it, or perhaps even work on sequencing the entire genome. That is great.
Awesome.
Oh, it's so soft.
It is very soft.
Yeah, it's even softer than I thought. But this is...
I need to squish it a little bit. When it gets here, if you want to take my gloved hand and just give it a good squish. Yeah, I'm probably going to get spicules in my fingers. I don't want to do that. That hurts.
Overall growth form is going to be, partly important for the taxonomic description, but then also these spicules that are on the outer surface of it. And then in hexactinellid taxonomy, they look at the spicules on the outer surface, on the inner surface, and then between the surfaces. And they're often, they're almost always different one from the next.
And they can also grow in different directions, too, can't they? And aren't hexactinellids named that because they have six-sided, star-shaped
Yeah, they're sort of like a jack, if you remember that. So it's like...
like jacks, like the 1950s toys. Yeah.
390-degree axes, And they... some of the very smallest, they have obviously some very large spicules that are making up the base, the stalk, and then other spicules which can be, they're microscopic, extremely tiny, and they make all these beautiful, like star shapes, and even with really interesting, little bumps and widgets on the end and stuff, and whether they attack...
Little barbells, like little barbells.
Some of them have barbells as well. Exactly.
They're really neat. They look like little pieces of, almost like someone took blew a little tiny piece of glass.
Yeah.
Like blown glass under your microscope.
So what I would guess is that this is in a group where, like Bolosoma, but that really is a guess at this point.
And this will go 95, not do formalin for this guy.
We don't need
He's
formalin.
He's going to be 95, okay. And where are they headed with this? Port side, okay.
Port.
That's number 4B.
I think that's going in the outside.
Port outer. Okay. Port outer's number 4.
It's just such a lovely animal.
I was watching, what was I watching? I think it was maybe it was like a Smithsonian show or a PBS show, and they took the sponge and they just kind of like squished it a little bit so that all the cells kind of came out, and then they watched it under a microscope and they started to reform. And I was like, oh my god,
yeah,
that's amazing.
Sponges have some amazing cellular biology. It's true. And this group of sponges, the glass sponges, one of the interesting things about their cellular structure is that a lot of their bodies are made up of giant multi-nucleate, what we call a syncytium. So rather than like a single cell with a single nucleus, it's sort of a massive stretched out cell with many nuclei all through there. Yeah. And they have cytoplasmic streaming, which is something I think that we see in plants and stuff like that. So
Caulerpa, which is the shallow water algae, is the same, right? Where it's one single cell, but it can grow like hundreds of feet long. And it has, of course, millions of nucleus. It's awesome. Will it grow back from that stem?
It will likely not grow back from that stem.
But if you were to rip the top off and let that float, that might grow back.
I suppose
it could settle
that's
down.
Yeah, I'm really not sure how good the regenerative abilities are of from that level of damage.
Oh, yeah, because we took the whole thing.
Yeah. Success.
Hey, Stephanie.
Hey, John.
Hey, I've been on a faculty Zoom meeting trying to watch this at once. I'm not sure if anybody has figured out what that was.
Do you know what it is?
I'm looking through our deep sea coral photo album that we've made, Christina made, and it looks very much like rhabdopectella tinnitus.
Abdopectella?
Abdopectella tinnitus. Yeah, it has a big head on it, a very thin stalk, kind of warty.
Oh, neat.
Round the edge. So I'll type it in, but
yes.
You can check.
Thank you very much if you type that in. All right, we're off to find something new. Oh, yeah, the big fan.
That big one out there at noon.
That's right.
Calling us.
We've had this target.
That is huge. Very candelabra shaped. That is a big, big gorg. What are you thinking? From this distance, I'm going to guess bamboo coral, just based on the rings at the base.
To see some.
So that's at least a meter. Nice.
All right. Get a look at these.
Oh, rhabdoplectella.
Oh.
Hey, sorry I've been on a call, but I saw this, and this is really exciting. So this is a bamboo coral. This is, we'll have to see it from another angle, but I think this is the candelabrum that Les Watling and I are working on describing right now.
Oh, nice.
Well, that's great.
Yeah, so this would be a new genus. If it's what I think it is, it's what we call clade I4.
Okay.
You're being very careful to make sure you don't ruin the name by using it.
Yeah, I don't want to the name. Yeah, no nominatums here.
Mm-hmm.
So what we'll be looking for is to see where it all comes together. It should be like one stalk that comes off the ground, and then you get a point where the branches sort of spring out. And is it a trident in the center there with branches extending from either side of the trident?
Okay, so you want a good look at the main stalk.
Yeah, the main stalk where the branches start.
Okay.
Great view. We're going to be able to look at this also to learn something about the sclerites, which are these small calcium carbonate microscopic bits that are embedded in the soft tissue that help create support and possibly protection against some predators, although there are plenty of predators of octocorals that seem to chew right through them. But those are very widely used in the taxonomy of octocorals, both for naming a species and for identifying them later. Mm-hmm. So this exquisite video that we get from the Okeanos Explorer helps us with that to some degree. Ultimately, you need to have it under a microscope.
Mm-hmm.
Very interesting, if people realized what is used to identify these things, and if you were looking at, like a field guide to the corals, most of it would be filled with scanning electron microscope images of microscopic bits and not the level we're looking at here, which is why it's so hard for us to make an identification when we see it on video. We're still working at combining those two elements together.
Mm-hmm.
Right.
Same with the sponges. I mean, they look. Some of them look exactly the same, and they're in totally different orders, and it's crazy.
So one of the things you see as we're panning down here is that these upright vertical branches don't have sub-branches. So that's one of the clues. Mm-hmm. Instead, it'll be like a candelabrum. They're all coming off of kind of like a base of a laterally extending branch, so to speak. And the older they get, the more of those vertical branches they add on to the sides.
Nice.
Yeah.
Mm-hmm.
And you can very clearly see the white calcareous internodes and then those black proteinaceous nodes through the tissue.
That's indicative of bamboo coral.
Yeah, so these ones, I do not see any of the needle-like sclerites poking between the tentacles, what we call intertentacular sclerites. They're probably there, they're just not so prominent as to be poking up between them. All right, so there's the middle point. That looks like a triple junction, doesn't it?
Yes, it does.
That's what I'm looking for. Beautiful, beautiful. This is it. What a great find.
Nice.
Thank you.
Yep.
Really, really nice. Mm-hmm. And did we get the lasers on that? I think they were, yeah.
Yeah. It almost looks like a bird leg with a stocking on it.
Yeah, it looks a little wrinkled.
Yeah, when you see them with these sort of thickened and swollen nodes, you know they're really quite old. And what's happening is the deposition of the calcium by the soft tissue is starting to grow over that proteinaceous node. So the farther down we get, the more it looks like a ring that's curling over. It's almost like, you know, when you see trees that grow into like a barbed wire fence that's next to it, it kind of girdles and grows around it eventually. So eventually, if we came back in another 50 years, we might not see a node there at all, but we'd still see that knobby knee swelling.
We're close up on that stalk. We also saw ubiquitous hydroids, which are coating the surfaces of everything, looking for small prey.
And can you paint it with lasers when you get a chance, just so we can get a good size?
Yeah, what a great view. It almost looks like it's one half of the candelabrum is growing really well, but the part closer to us is not. That part has been broken off. And I'd say that this colony has been stable on that rock for a very long period of time.
Yeah, for sure.
Yeah, you can see the blue sponges growing on the rocks there behind as well.
All right, here come lasers. Yeah, so lasers are 10 centimeters.
Wow.
So the candelabra part at the base is at least a meter. So that would put that stalk at another meter.
2 meters tall at least.
Yeah.
If you can, it's not terribly critical, but if you can, when you're moving, if you could get a face-on view of that candelabrum, that would be so awesome. But
so you want us to swing? You want us to swing left, so we can get a view from the, like a more of a front facial. Oh my God, I cannot talk today. Yeah,
a frontal view.
A frontal view. Left, our side, 9 o'clock. Yeah, it's like swing 9. So we can see all the individual branches.
I think here, if we get that, you'll see that it's going to be lopsided. So it's growing off to the left, but not to the right. It should be adding those vertical branches symmetrically around the trident. The center part where the branches appear to begin.
Awesome.
Very nice. So we're still below 1,400 meters.
Yeah, we haven't moved very much at all. We got somebody up there.
And you know, what we're seeing is really characteristic of 1,400 meters. So this is looking a lot more like, you know, offshore seamount at this depth than it is when we're inshore on the Blake Plateau and seeing the Lophelia mounds. We're getting a different community, a different representative group of corals.
Absolutely.
Really, thanks so much for that imagery. That was exquisite.
Yeah. We're good whenever you guys are.
It's just so much to look at. Not sure what's going on.
One more pirouette. We're going to go around the other side.
So to the left there might be another bunch of those hermit crabs again on a dead skeleton. Isn't that interesting?
Yeah. Similar to the one before.
So I'm curious if this mound is not biogenic, which it does not appear to be, although we've still got a ways to go to get to the top.
Mm-hmm.
But then how it might have formed.
I have no idea.
Yeah. Maybe there'll be some geologists in the chat
You
who
can chime in and let us know.
could either call in or type in.
Oh, neat. Here's the backside of it.
Mm-hmm.
Kind of a cool view. You can see that all the polyps are concentrated on one face.
Mm-hmm. I would assume that face is
Question is
facing the current.
Well, I was just going to say, Stephanie, is that the downstream or the upstream
Yeah.
face? So in some of them, it's downstream because the water flows through and the baffling of the branches causes there to be these little ripples behind it. And so maybe it's better to be on the downstream side
Right.
than the upstream side. I'm not actually sure what it is for this one. Great view here, though. As we are zooming in, I can really nicely see the original trident point. And here you can see what I mean about each subsequent vertical branch starts as, this curvature from the existing one at the next node. And normally that would be symmetrical around the original trident starting point. And the hypothesis here, we haven't been able to prove it yet because we haven't had the appropriate samples, but the hypothesis is that when it's really young, it's growing as a whip. You see that it has to get to a certain size before it starts putting out these side branches.
Right.
I've never seen one that's really short to the ground and grows this way. So they all, have that center stalk grow for, let's say, 50 centimeters to a meter before they start branching. Just incredible.
Awesome.
Good stuff.
Yeah. Really good.
There's some more Stigopathes at 9 and at noon. Looks like we have some undercut ledges a little bit.
Another Metallogorgia.
Mm-hmm.
A Metallogorgia.
Metallogorgia?
Yes.
So the next thing we have to train you for is finding what we call the teenage Metallogorgia, because they only have that sort of terminal parasol as their final growth stage. As they're growing, they have branches come out along the stalk, and they don't have anything at the top. It looks like just a little antenna. But then what happens, we don't know why, but when they get to this phase, they drop all the branches along the stalk.
That's interesting. Looks like there might be something dead here at 3. And then in the center we've got a
Is that a dead whip?
ophiuroids all
With
over
lots of
something.
ophiuroids?
All over a whip or...
I'll bet it's a dead Metallogorgia.
Oh, dead Metallogorgia. Yeah, you can see the branch
Yeah.
hanging at the top.
Right, and so this is instructive also because you see it doesn't have the Ophiuchreus, which is the brittle star that lives associated with it. And on the live ones that did have the Ophiuchreus and the purple tissue on it, you didn't see anything on the stalk.
Mm-hmm.
And so they tend to keep associates off of it. And I don't know if that's just the Ophiuchreus ophiuroid that's living there to sort of swats things off from settling on it, or if it's some secondary chemical that the coral is producing that is noxious to anything settling on it. But you only ever see stuff on the dead skeletons. So very instructive there.
Neat. Okay, so it looks like these rocks are starting to increase in more like a piling of boulders.
Mm-hmm.
They're about 1 to maybe 2 meters wide, maybe half a meter tall, with sand in between. A slight rise here. A very slow slope coming up, maybe 10 degrees looking ahead. Another bamboo ahead, it looks like. Different species here. Oh, there's something in the water. Quick,
Yeah.
idea, quick.
It had tentacles. You can really see the current moving across.
There's another bamboo, I think. Maybe not. Might be something else.
This is the Primnoidae again.
Primnoid, okay.
This one may be different from the one we looked at earlier, and I'm basing that on the orientation of the polyps pointing downwards, where I believe last time they were pointing upwards. So this could potentially be the genus Narella, but I'm going out on a limb there. Let Alan talk about the hydroids there. Alan, is there any hope of identifying hydroids in situ, or is it just too difficult?
Yeah, probably too difficult, other than, maybe getting to a family or something like that.
Yeah, there's something yellow here at 10-ish. And this looks like a bamboo coral at 2-ish, the tall one. Yeah, that dead center pink thing, that'd be cool.
Suggested flying by.
Maybe this is Bathypathes, maybe? Yeah. So that's maybe Bathypathes bipennata. Type of Antipatharian. That's my best guess on that guy.
Yeah, this looks like Bathypathes, a different kind of black coral, a different genus now.
I got it
All
right.
crowded in together.
Yep. I like how all the black coral generic names seem to...
Pathes.
Pathes,
Yeah.
yes.
Makes the life a little easier when you're learning Latin. So they thought they...
Yeah, and I guess the factoid, we have to give this factoid out every expedition, but they all belong to the Antipatharia, and Antipatharia translates as against disease, and it's based on this old Greek idea, 2,000 years ago, that crushing up the skeleton would have medicinal properties, which it does not. That's where the name comes from.
There is a really cool spiky ball right off screen when you guys are done looking at the Bathypathes. It's right above, like at 1 o'clock, right off screen. There's this really weird spiky ball. Say it's at noon. Just popped in.
And I think the white thing there next to the black corals, that's a corallium that's standing upright. We looked at one that was knocked over before. Or hemicorallium, corallium.
There's also a single-stalked Antipatharian here.
Hermit crab climbing up on top of that.
Same pink hermit crab.
Corallid.
And there's also some kind of legged thing, pink, maybe a sea spider, pink sea spider in there. I think that's what it is. I see his little legs.
I wonder if it's feeding on that corallid. Be nice to see it
Munching.
It munching, exactly.
Munch.
No offense to the corallid.
Oh, yeah, it's not personal. And maybe a Chrysogorgia, too, at 3.
Great. Wow. Huge rocks.
Yeah, this is a big boulder. Maybe 5 meters wide.
This is so interesting to see this community, because if I didn't know better, I would think I was on an offshore seamount and not on a sedimented continental slope.
Mm-hmm. There we go.
Oh, yes.
Too cool.
You can see it. It's got its
What, proboscis?
proboscis there, and it sucks the juices out.
So there's pink on this coral. Is that coming from it being traumatized?
I don't
From
know.
being
That's
eaten?
an anemone.
It's
an anemone.
Oh,
growing on the
anemone.
coral.
Okay.
And it's unfortunately been attracted, or it has attracted this sea spider to suck its juices out.
That is such a cool sea spider. He's bright pink.
Yep, filling up its branched stomach, which descends down into its legs.
Yeah, very cool. I don't know how many people realize, Alan, you described that well, the sea spider and that long proboscis and the suction, but most spiders suck their food. They liquefy their food before they ingest it. And the sea spider isn't that closely related to terrestrial spiders, but terrestrial spiders do something similar. They basically pre-digest outside the body and then suck it up.
That's really cool. Predation caught. Now I wish we knew more about that anemone and what it actually looked like when it's out and happy, so we have a more specific. And all these hermit crabs are climbing up on things.
You got something neat in that hole up there.
Yeah.
Might be a crab or something. And we have that spiky ball too up here. That one. It's tiny, but. Oh, that looks like a coral or something? Maybe a solitary coral?
No, it's an
Or
Octa
two.
Coral.
Oh, yeah, it has 2 polyps. Maybe a pink Mesodacea or sponge above it?
It's probably Anthomastus or
Anthomastus.
related to Anthomastus, Pseudanthomastus, something like that, which is an Octa Coral, which does have these relatively large polyps. They kind of grow a little bit like a toadstool, and the polyps would normally be coming out of that, so this would be an early stage, if that's what it is. It's very unusual. Actually, yeah, there are 2 polyps there, aren't there? So that's almost surely what it is, just a relatively recent recruit.
And these yellow hexactinellid, maybe?
No, definitely
you know?
Devon sponge. Yeah.
I think these one too.
Yeah. The orange demo sponge, and then tan right to the right of that, and then on your way to your spiky ball. Which,
I don't have a clue what that spiky ball is. Sponge.
yeah, I'm thinking that's a sponge.
Baby sponge. Maybe a hexactinellid in front of it.
Possibly a small. Yeah.
That's neat. Little starburst sponge.
Really great.
Okay, we're good whenever you guys are. You can see the dead stalks here. Ooh, we got a squat lobster at noon. And then we had a yellow sponge to the... Okay, pilot change, no problem. Mm-hmm. Yeah, there's a lot of stuff down here.
Yeah, we're still at 1,400 meters.
We haven't moved at all. How much longer do we have on this dive? Let's see, 3:45? 2 more hours. So the goal is to get to the top in the next 2 hours.
Yeah.
We got plenty of time.
An hour and a half to get to the top.
What, John?
I think you have an hour and a half to get to the top.
Hour and a half, yeah.
You got to be.
You got to be off bottom at
We got to get up there.
5:30 or 5:45?
I think it's
5:30? 5:45 we're coming up?
5:45.
Yeah, this yellow thing out. Yeah, there's a white thing in the background at 9. I'd like to take a peek at. We're just not going to go. Just sit down. We're just going to stay here. Just look at all this stuff on this one rock.
It's just so rich, you know. It's not really dense, you know, like you see with the coral mounds, but there's so many different things, and there's at a lot of different scales.
Mm-hmm.
It's some.
I noted before, since we're sitting here, that bamboo coral to the right is different from that candelabrum that we were looking at.
Oh, yeah,
earlier.
definitely. It's totally branched different. It's internodal branching, too, whereas the other one was nodal, right?
Hi, yeah, excellent. So it's probably the genus Keratoisis in the genetic clade D. Had a tough life. It's missing all the tissue from the lower part of the colony. So
Yeah, right.
may have had
Looks pretty beat up.
encounter with a sponge at some point. With a sea star.
Mm-hmm.
Star at some point.
But live to tell the tale.
Mm-hmm.
Via.
Yeah. We actually have our eyes on something at 9 o'clock when you guys are done, so don't drift too far away, please. Hey, Scott, can you type Keratoisis into the chat for me? It's Worms hates me. So, yeah. Yeah, this yellow sponge we are after right here at 6 o'clock.
Keera.
Thank you. Oh, Kera.
Yeah.
And I see a sea star, I think, right below it. Maybe a goniaster. So this yellow sponge, it's got lots of film on it.
Yeah, that's
Like a bacterial mat on top. Kind of sickly. It's pretty neat, though.
yeah. It may like it like that.
Yeah.
You know, we often see what we refer to as these deep-sea spider webs. We find them on overhangs and so on.
Mm-hmm.
And just recently I saw a paper that I goes, oh, maybe this is what I'm seeing. And that is the sperm release from some of these sponges comes out with all this mucus. I'll try to find a link and send it to you, but Alan, you may be familiar with it. And I'm wondering if that's what a lot of this sort of webby stuff is, evidence that there's been this spawning from these sponges.
Possible. This almost looks like it has a fistule on top here. This, like, sharp. I think that's part of the sponge.
I would guess not, but I really don't know.
Yeah, that's neat.
What is the structure that the sponge is growing
I think
on?
it's a dead coral.
Oh, okay.
That's a guess. Old dead. It's very old. You think? Like, it might be a dead old sponge. Yeah, and then we've got this little star here.
Mm-hmm.
It's also a single-stalked gorgonian of some type.
Good eyes and another
that light's a
small
little
white
better,
sponge on the left-hand side there.
Are we putting this in the demo or the hex?
That's definitely a demo sponge.
Okay. Maybe a worm tube? Yeah, that looks like a dead coral.
I'm saying a sponge stock.
Sponge stock. Is that another hermit out there?
Yeah, another pink
All
hermit.
these lots of hydroids colonized on this dead coral.
For the base, not that.
No, I thought there was something in the distance.
Like tree branches.
Yeah, it's crazy. Yeah, we can move on, I think. We got a good look at most of this stuff.
Yeah, we got to keep moving.
Not too fast, though. We got 2 hours to kill.
That's true.
And like, I don't know, 100 feet to go. How far is this?
We can still rise another 70 meters or so
Okay.
to get to the top of this. Do you think this is
white
a
ball's
convenient time? Yes, this white ball is great and would be great to get a zoom on it. That we should get our middle of our transect eDNA sometime soon.
What was the depth? Why don't we wait till 50 meters up?
Okay.
Because it's a 100-meter difference, right? So if we came up 50 meters and then did it at the top, 100 meters.
Right. So we'll want to do it at about 1380.
Yeah.
What do we got here?
Ooh, Oceanapia, maybe? Oh, yeah.
Yeah.
I guess is Oceanapia on that guy.
Right
but
next to a Pharea, a glass sponge in the background, but this is, wow, just a
That's
beautiful
super neat.
demo sponge. The morphology you don't see often.
So I didn't catch the name. This is the thing I always call polymastia.
Oh, polymastia. You're right.
That spiky one we saw earlier was a small version of it. So what was the name?
Polymastia. You're correct. That's right. I was thinking Oceanapia, but you're right. It's polymastia.
I'm going to have to trust you guys on that one.
Don't usually come up with the correct sponge name. So that's
Oh, yeah, no.
Thank you.
You're right. We got some worms, too. Some feather dusters here right off screen, right at the edge of the screen.
And a nice blue sponge tucked underneath that guy.
Oh, there's some Sargassum detritus. So that's just drifted down from the surface. Maybe an anemone. Yeah. Again, it's like this pile of boulders with sand in between. The boulders are 2-3 meters tall, maybe. I mean, wide, and maybe a meter tall in parts. They're kind of jumbled, like a pile of rocks.
So I just heard in the chat that Bruce Mundy ID'd one of the fishes that we observed yesterday that we were unable to identify as a juvenile lancet fish.
Oh, cool.
And that was a very unusual observation. So
nice.
It was very nice that we were able to capture that. It went out, and people were able to use that high definition video and to create more knowledge.
Yep. Give us some feedback.
It's perfect example of this telepresence and how well it works in these NOAA ocean exploration expeditions.
Yep, community engagement. There's a little white stalk here at the center. It's probably something has passed, dead. Another Stichopathes, some dead stalks.
See these big piles of boulders. And I always want to know where they came from.
I know.
Where did you come from?
It looks like, 'cause when we're in the MPA with the same kind of similar, like, squarish boulders, and they look like almost like they were formed from the pavement and then underneath collapsed.
That's nice. Get a
Yeah,
little
a rat tail.
A little.
Nizumia, maybe?
Really nice. I think there's is that a gastropod on that dead whip? Oh no, that's another
A hermit.
another hermit that's climbed up, and another. But there's a fish to be seen.
Mm-hmm.
Oh, so cool. More Metallogorgia.
Yeah, it looks like that bamboo coral's dead. There's just the stalk left.
Yeah, and I think that's an candelabrum one as well. You can see the trident in the center again, so it's informative.
Mm-hmm.
When I say informative, what I mean is we're still learning about the distribution. We want to know, where it's found. We've found colonies in the Pacific and the Atlantic, and they seem to be within a certain latitudinal zone, so tropical to temperate, but not further north.
At 3 o'clock, we have a fish in the sand. And then above that, we're going to have something yellow on the rocks.
Who's joined us on the line?
What this guy is. Oh, he's neat.
He is. Very beefy eel.
He looks kind of beat up. Like his skin's sloughed, right?
Mm-hmm.
I don't know if that's normal or not.
This will be someone's.
Cuskill, maybe? Oh, he's got little feelers.
Yeah, he does.
He probably doesn't see very well. It's pretty deep.
So, is this a conger eel or a cusk eel? I don't know.
It's a fish. That's the best I can do. It's a ray finned fish. That's where I tap out.
That's all right. Great footage of it. Should be very easy to identify for someone who knows what they're doing.
Look at that beautiful sponge at noon.
Oh yeah, let's check that guy out. Cusk eel is what.
Cuskill? Okay.
Well, that's what Scott wagers.
Oh, here we go. Kevin said it has feelers in the... or it's pelvic rays. That's neat.
Chidiformes.
Ophidoformes.
Ohidoformes.
Ohidoformes.
There we are. Ohidiformes.
And if we could glance to the left, Steve says there's a lyrate colony we haven't seen before.
I'm not sure. What kind of colony? Black coral colony? It might be what they mentioned. So yeah, behind the Sticopathes is this orange whip in the front, and then there's another black heart right behind it. It has gray polyps. Really loose branches. I think that's okay. It's that orange sponge I'm interested in.
Yeah, the black coral, I'm not sure if that's a young Leopathes.
Leo, okay.
Or if that's something else. It's definitely new for the day, though.
Nice.
Now we're going to come get a look at the sponge.
Oh, neat. It's like a skirt, like a fan skirt.
Oh, this could be the glass sponge Hertwiggia.
Looks like on top of that rock we got another flopped over coral too. There we go. This is bright yellow. And you see the veining in it?
Yeah. Yeah, I don't think that's Hertwiggia. I think that's a demo sponge.
Nice.
Very nice footage.
So this is a yellow, I'm just going to call it a yellow skirt sponge. Or amphitheater. It has veins. All right. Sounds good to me.
Thanks for getting that. Nice view of that yellow sponge.
So again, we're looking up slope here.
Okay.
Our midway point is going to be about, what, 1380?
Yeah, about 1380, in which
Okay.
case we'll take another
Water
water sample.
I got something here as a fish, maybe? Another rat tail at 6 o'clock. Oh, he's in the breeze. Oh, Lemonema. A codling of some kind. Look, there's Feria in there, too. Yeah, we're good. Looks like a funky sponge here at 6. Possibly dead. Can we look at that coral at 6 o'clock? I think there's something in it.
Is that trash?
I hope not.
Me too, but it would be good to take a look.
Yeah. It's going off screen. There's a coral going right off the corner screen, yep, on the right. And it looks like maybe black trash inside that coral head. I just wanted to get a better peek. I also think I see a giant star in the background. It looks like trash to me. Ugh. That looks like plastic.
Very sad to see.
Ooh, there's a cool, right underneath it, there's a giant crinoid. And then off in the distance at like 3, there's a different starfish or sea star. There we go. Now this one looks similar to the other one we saw, but this one doesn't have those long pinnules at the tips. I think the other one was Analceta metra. This one looks like a different species. I think there's 10 arms. Hi, who chimed in?
Just me again.
Hi, John. Neat. So this is a type of cumatulid. Sea lily is the common name, right?
Sea lily, yeah.
And if Chuck Messing was on the phone, he'd be able to tell us from the footage what it is. But
it's a beautiful crinoid, is what it is.
yeah, it's pretty cool. So they reach their arms out and they catch food in their pinnules, and then they pass it from one pinnule to the next, all the way down into their mouth. And their mouth is right there in the center of that arm, those arms, just like most echinoderms, right, relative to the sea stars. I was actually watching something, again, that PBS special, right? I was watching that PBS special where they were talking about how bilateral symmetry is actually what gave rise to all the ability for, the evolution to take place, right? So lots of variety in that bilateral symmetry. And the oldest echinoderms that we have fossils for were actually bilateral, and they formed radial symmetry over time.
Yeah.
So that was pretty neat.
There are.
What is a pinnule?
Yeah. As someone who
What's
studies...
the pinnules? The pinnules are the little arms off the arm. It's like the subarm, right? So, sorry, go ahead. I
No, I was just going to make a comment about how there's a lot of diversity in the non-bilaterian animals as well, but I get the point.
Yeah, because like the first animal fossil we have, the animal actually looks like a plant. Like it's a long stem, and then it's almost fractal.
Yeah.
Right? Where the tissue fractals into these. Yeah, you really have to just watch it.
The
Super cool.
Ediacaran fauna. Yeah.
I got a sea star coming up. My guess is some type of goniasterid. And a little blue sponge at 3. And again, another Anthomastus past that at 3 o'clock. Yeah, let's get a good look at this sea star. Yeah. Some type of goniasterid.
I'm falling down and I can't get up.
I know, right? It's probably, yeah. Looks like it had lost an arm maybe, regrowing an arm at the top. It's a little shorter than everybody else. Could be eating. Maybe eating that primnoid it's on. It looks like the tips of his arms have like almost like a forked hook.
Yeah.
That's cool. So goniasterity. We're good whenever you guys are.
Keep your eyes open for a dead Lophelia. John Reed saw one.
Oh, where?
Yeah, it was several minutes ago.
Okay.
So we're a little past.
No more than 10, 15 centimeters.
Oh, little guy.
It looked like the typical old dead black Lophelia.
Okay. Was it standing?
Branching, I could see polyps, but we were beyond it before I had my phone died on me, so I wasn't able to call in right. So anyway, it appears to be around here.
Okay, I'll keep an eye out. Looks like we have a couple fan sponges and some maybe white corals, some gorgonians. Now that looks like Ficalia to me. See the veining?
Yeah,
so
the
veining,
top one.
Yeah, the veining on this might be on the other side. If you face looking at its coming in from the right, there might be some fanning. Oh, it looks like a sea pen on a rock in the background too.
Good eye, Stephanie.
Now you were saying these are important. You want to collect that?
So, I mean, that would be so fantastic. We should get a good image of it at least. What I can tell you is that there's only a single, maybe there's two now, because maybe Okeanos collected one 2 years ago, but as far as I remember, there's only a single documented collected rock pen in the Atlantic. All the others are in the Pacific. And that one that was collected in the Atlantic was collected in the Bahamas on an expedition we did back in 2009 and is a species known only from the Atlantic, and it's the one that's in a different genus from all the others. And when I was discussing yesterday why that collection was so important was to ascertain, you could see, doesn't this look a lot like the second sea we collected yesterday?
Yeah.
Mm-hmm. Sediment? Yes, that's what we were saying, that overall colony morphology looks similar, but here you can see that it's kind of like a bathroom plunger or suction cup, the way it's held onto the rock. The peduncle has been modified to cling onto it in that way. So that's what's different about the rock pen.
So this we would call this a collectible.
I would.
Yeah. Can we collect this? Can you guys get in there deep enough to collect that?
So if you're going to consider that, let me say that it can be grasped in, the Tygon tubing part of the claw, and then just kind of wiggled, and it'll pop free of the sediment.
Okay.
Pop free of the rock, and it'll come off pretty easily. It doesn't need to be cut.
Okay.
That peduncle is critical. We really like to see that.
Yeah. Did you guys hear what Scott said, or should I repeat for you? Okay. So the rubber tubes inside your claw. If you can just gently, without actually cutting it, just gently squeeze them with the rubber part and then give it a little wiggle, he might just pop off that rock, because the bottom part, the peduncle part, is actually the part that's really important to maintain the structure. Again, you see this inside that fan at 9. You're getting that veining.
Mm-hmm.
So that's probably Ficalia. This is going to be sample number 5. If you can get it. You don't think you can get in there? Maybe a chrysogorgid there at 9 o'clock, too. That furry. Okay.
Yeah.
Yeah, if you guys can't get it, it's fine.
If it's not collectible, it's okay. He's really happy with the footage. Up until yesterday, I did not know of the existence of rock pens.
Yeah, because they're always in the sediment.
Yes.
So this is the one weirdo from the family. This is the black sheep. Mm-hmm. You can see the inside. Mm-hmm. It's like tissue. Almost muscular, like that.
Oh,
like fibrous.
Yeah. It looks like the part that's that's under the sand, usually only.
Yeah, you don't usually get to see it because it's underneath that,
And then
that
it's
sediment.
modified to have this adhesive foot. It's pretty neat adaptation.
Alan, you said adhesive. I mean, I don't know that it's not adhesive, but the descriptions based on the collected samples in the Pacific suggest that in the center there, you can see at the bottom of that rod, they use musculature to pull up the middle center of the underside
like
of
suction.
the peduncle to create that suction.
Hey, Scott, do they move around?
That's great.
Not to my knowledge.
Usually once they're down,
I've
they're...
also tried them. You know those clowns that you can sort of box with that sort of fall over and then bounce back up? I've described them that way. So we have seen them become dislodged and they drift for a while, and I suppose they can hopefully upright themselves.
reattach.
But, I mean, they're not mobile in there.
It's not like an anemone that'll kind of like crawl around the ground, you know.
Not to my knowledge, but
Okay.
you know, we don't know all that much about them. There aren't all that many observations. I will say that we found an observation in the Pacific not too far from Hawaii at about 350 meters where we saw these huge fields of them, literally thousands of them. And that was just an incredible sight.
Nice.
But otherwise, yeah, we normally just see them in 1s, 2s, and so on. I say normally, like we've seen a lot. We have not seen that many.
Well, good eye spotting that guy.
Yeah, he's pretty small. Move the rock first? Yeah. See if that's loose.
Can you get the whole rock?
That's what I was thinking. Just grab the whole rock. Stick it in the rock box. I'm not sure, John. It looks like that rock's pretty solidified in there. Yeah, the boulder.
Do we have a taxonomic name for this collection?
Besides Pennatulacea?
Yeah. Thinking that Upsana says in the chat maybe Anthoptilum or Calibolemnon.
Yeah, Anthoptilum or Calibolemnon. So the one in the Atlantic is currently Calibolemnon,
Okay.
and the ones in the Pacific are Anthoptilum. So your choice.
Great. We're going to go with the Atlantic name, right?
Yeah, that's what I put in.
Thanks for that, Scott and Upsana. Calibolemnon, I like it.
Looks like a little wishing bone behind there. Little rock, little branch thing. See it? Little deep sea wishing bone.
Yeah. Oh, it is small.
Yeah, it's pretty small. It's like 10 centimeters long, maybe. Yeah, just gentle, yeah. Whoop! Popped right off. There you go. You got it.
Nice.
There you go. Peduncle's intact. Look at the little ring it left. It had to have been there for a while, huh?
Yeah.
That's pretty neat. Good close-up shot of the peduncle. Beautiful. See, you can see it like turns into a suction cup, right? It
Yeah.
sticks to that rock.
That's a really cool way.
Like a bathroom toy. A bathtub toy.
That is such a good view. That is not a view that we get in situ ever. Fantastic. Nice work, pilots.
It's a sea pen. It's a rock sea pen.
Oh, was that a...
Yeah, the peduncle.
Now we get to watch it go into its container. Port inner.
That one is going to be number 5, yeah.
Great, 5B. Nicely done. It's in the box.
So I was just realizing the increased value, particularly of this specimen, relative to what we did yesterday, because these are two locations that are quite close to one another. So this may be the first instance where we have, within 30, 40 miles of each other, two colonies that look the same, one in sediment and one in the rock.
Yeah, cool.
So that's going to be really interesting to compare.
Awesome. Thank you.
Oh, and I just see in the chat the Calibelemnon species in the Atlantic is named after you, Scott? Is it Calibelemnon franci? Yeah. That's quite an honor.
Yeah, that was a real surprise. Gary Williams, Cal Academy.
That's great. Nice collection.
Looks like a white gorg noon, or 1. And a little yellow one next to it. There might be another Bathopathes in the background. Yeah, some kind of Gorgonian. This one's pretty disorganized.
Mm-hmm.
Planar.
Is that Huh.
Yeah, I'm going to go again with Coralliidae for this one. Hemicorallium or Corallium. I'm not sure if Steve Oskevich is still on. He might have something to say, but that kind of looks like what it is. I'm seeing lots of these smaller bumps behind the polyps. Those might be, what's the word, siphonozoids, a secondary polyp that is non-feeding, really small for funneling water.
Mm.
It's either that or it's an Anthothelidae, but I'm pretty sure it's Coralliidae, in which case it would have a very hard, brittle skeleton.
Mm-hmm.
Man, I'm learning so much on this.
Yeah, see these white, this like clear sponge here on the rock that we see down here?
Mm-hmm.
That's what I call an ice sponge. I have no idea what else it is. And they're like impossible to get because they're so thin.
Swiftia poking up from behind it on the right, those red, slender red branches. I think that's another Octa Coral.
The Swiftia?
To the right of this, I believe.
Okay. Okay.
Oh, yeah.
Oh, yeah.
Just peeking up behind the rock.
There's also this yellow.
Is that yellow? Do you know? Is that
I don't know.
Another Gorgonian?
That's Paramaricia for
Paramaricia
sure.
for sure.
Classic planar
Paramaricia?
shape to the colony.
Mm-hmm.
And then with that one brittle star hanging on it, classic.
You said Paramaricia?
Yes, I did.
Okay, thank you. Or Paramaricia.
Now that's one of your Aspire targets for connectivity, right? The Paramaricia plus the brittle star?
I believe so.
That's correct, Alan. There's several species. It's really hard to tell these species apart, even in hand, and even the genetics. It's really subtle, but they're very widely distributed, a very common genus, and so yes, it is a Aspire target.
So would we like to add this to our collection while we're here looking at it?
Sir collects a lot.
I'll leave it to you guys. Alan, you're right. This is one of the ones that Andrea identified.
Yeah, and I saw it on the Aspire list. I don't know. I think it would be smart. We haven't seen very many of them.
Yeah, we can get it.
Yeah.
I'm fine with that.
Yes, Pilot, if we could collect these two together, that would be terrific.
I would imagine we're going to have to... Are we going to jar them together?
They take difficult to take apart?
Yeah, you're either going to have to
I would just jar them together.
Yeah, just jar them together, because we're going to have to cut that ophiuroid out, and he's still going to have it be attached to those. There's no way we're getting him off that Gorgonian.
Yeah, that's what I figured.
Yeah. I mean, you can. It takes a little while. It might be worth snipping the tip of one of the arms.
Oh, yeah, no, we'll definitely take a separate DNA. But as far as the actual sample in the jar, I would like to keep them together in the same jar, and we'll just get 2 DNA samples from them. I think that'll work.
It depends how much time. They can be separated. But I did want to say, you can clip the branch below where the coral is, where the sea star is, if you want to try
and put him in his own jar. Yeah.
And this is, it needs to be cut. You can't just snap it. So this is a cutter, not a snapper.
Hey, pilots, this is going to be really strong to the rock, so your best bet is to use your snippers and clip him below, near the base, if you can get down that deep. Thank you. Correct. Biobox, yeah. I would think he's too big for a suction jar, but... This is going to be sample number 6.
And this is for connectivity study.
Oh. This is number 6. Hey, John, I think I can hear you typing. Thanks.
Nicely done. That is just...
awesome. Look, and we left a stem, so maybe he'll regrow.
Only if he finds another brittle star. Or another brittle star finds it. So these samples that are targeted for connectivity, it's to help understand, how these deep water communities are connected across distance.
Genetically,
exactly.
Yeah. We have that constant flow of the Gulf Stream, right? That starts up in the loop current in the Gulf of Mexico and then comes around the Straits of Florida and then up through the Gulf Stream, right? But we're way further east. So there's probably a lot of Bohemian water coming in this way.
Yeah. I think this is where the water from the Labrador Sea gets entrained onto the Blake Plateau. So
this is a good place to be mixing genetics.
Yeah.
Right? As we're going to get probably maybe two genetic populations, would be my guess. We got a bunch of stuff from the Gulf side and then a bunch of stuff coming up from the Atlantic. All right, and collection.
Well done.
Thank you very much for that. Oh, sorry, that was 6B,
6B, yep.
not 5B.
And we better think about taking some water sometime
I know.
soon.
Let's say 2. Yeah, right. About here. I need some water too.
Well, feel free.
Yeah, I'll go take a water break. Depends how far apart they were fired. We were right in the same spot when you tried to fire 2 and 3, but we don't. Do you know if 2 fired like a long time ago? We don't really know when 2 actually closed. Okay, I think we should toss 2 just because. Well,
Yeah.
we'll talk about it. We'll grab it, but let's just not count that because I already have it not counted.
Mm-hmm.
So what's in 3 I actually counted as sample number 2. So the parameter seed we just collected is number 6. And then the next water is going to be probably 7, unless we find something magical between now and the water.
And that would be a Niskin 4.
Yeah, and that's going to go in Niskin 4.
And we'll save the final one for up on the top of this mound.
Because we already got a 1,000-meter one, too, on the way down before. Ooh, look at that.
Yay. Little comb jelly floating by. Bathosiroi, I think.
Yeah, little shrimp next to it.
Yeah.
Ooh.
Or it's... Get back in on the chat, see if I'm...
There's another sea
missing
star. Look at
anything
that
over
at 2 on that rock up there. All right. Yeah, this is quite a slope. Maybe 20, 25 degrees here. Down slope, looking.
Yeah, it's so funny. You know, you look at this view and you think maybe there's not much living there. And then you get close and the lights are shining and all of a sudden you see that there's
tons of biota.
More things than you can...
Yeah, even really look at.
Yeah. We've come about halfway up this mound so far.
Yeah, I say call that water sample whenever you're ready. That's going to be number 7. Yeah, we're going to take that water sample when you're ready. We're about at the right depth for that. So that's going to be 7W.
Yep.
You just fired it? Is that what you said? Thank you.
Excellent. Thanks very much.
20:34:40 is the time. And that was at 13:84. We have another sea star here. Probably Goniasterid. Let's see. And get closer. Looks pretty deflated. It's kind of skinny. Neat. He looks like he's doing pretty good on his COVID diet. Nice. You can see his tube feet.
Really nice.
Yeah, that's cool. There's also a Feria down here, it looks like, underneath
Very
him.
nice glass sponge. Might
Maybe.
be dead. It's very
Yeah,
thin
could be.
and
Parts
fine.
of it are, right? That, like, it almost looks like burlap
Mm-hmm.
when they die. Hey, is this Chris?
Hey, it's Chris Ma.
I got it. I knew it was you.
Interesting.
Yeah, what do you got?
This is this is paradoxically one of the irritating things about goniasterids. The more nondescript and plain-looking they are, the more likely it is that they're not something that we have described.
Oh, of course.
I mean, the really distinctive ones just come right out at you, but it's always these kind of plain ones that are irritatingly difficult to pin on. This doesn't look like either Ceramaster or it might be Mediaster.
Mediaster?
Yeah, Mediaster. I can put that in the chat. But it doesn't have granules on the... Okay. We're at 1900. Yeah. Are you in a position to collect? I know you guys just did your water sample.
Stars are pretty quick.
Can you?
Yeah.
Yeah. Can we collect this, pilots, or is that we can try, Chris?
I mean, I understand, obviously, but if we can, that'd be great. Thank you.
So that's going to be sample number 8, if we can get them. Do you think you just undescribed species, likely? Is that what you think?
Yeah.
Okay.
Yeah. There's probably something it's feeding on underneath it, too. I mean, there's a whole bunch of, like, encrusting stuff on the rock there.
Do you think the suction hose is the
if you have
easiest?
to move on, I have to keep the timetable. That's fine.
It's fine. We don't have very far to go. Like, it's a short dive, like a short distance for us to cover.
Yeah.
So we've really been poking around a lot. Do you think the suction hose is the best way to get him? Yeah, it might be too big for the suction hose.
So this thing is about. So the lasers are what, 4 inches
10.
across, right?
Yeah, 10 centimeters.
So it's actually not that big.
It's not, but the suction hose is just too
it'll
small.
It'll come off pretty easily, I think.
Okay. You think so that we can grab him with the claw?
Yeah, that'll work too.
Okay. Oh, yeah, go for it. Sure, that works. Okay. Perfect. Perfect. Not live.
And as long as I've got you guys on the phone,
Yeah.
the one that you guys saw earlier, the white one, that was a sort of a familiar predator of corals, and it was actually feeding on. I'm not sure if it was a primnoid or something.
Paramyroseid, I
see
thought.
the vestiges of the skeleton in the back, but that was a Cerciaster americanus, which is thanks to Okeanos, now one of the better-known predators of Octa Corals down there, in addition to Evoplasoma and Typisteria.
What was the name of that guy? Can you repeat it, spell it a little bit, or put it in the chat for me?
Sure.
And
I actually put it in the log.
Okay.
But it's Circeaster.
Oh, I see it.
Spelled
Okay.
like goddess of magic, like Circe, Circeaster. This one is a goniaster that looks vaguely like Mediaster, but it doesn't quite fit.
Okay.
Fit the one that's supposed to be in this area. So
Great. And I forgot to note the genus of this.
Of this one?
So
close to Mediaster is the closest thing I could come up with.
And Mediaster is M-E...
like Mediaster.
Good. Mediaster.
Yes. The name literally translates to medium star, so...
That's pretty easy to remember.
Yeah.
All right, so he's going into suction. Great.
T05. This is sample number 8. Oh, yeah.
Yeah. It's attempted.
Don't cross the beam.
Looks like maybe.
I can see him at the end of the tube.
Mm-hmm.
Yeah, we can too. I think they're either going to try and suck him in or turn around and blow him out into a bio box. I'm not sure.
Sea Star saying no thank you.
How long is the suction hose?
How long is what, John?
The suction hose. How far out can they stretch that thing?
Oh, pretty far. I'm not sure. 6 feet. There you go. 6 feet from the ROV. Okay.
You know, we've got so many things stuck in these corrugated tubes. It's kind of tricky sometimes. Sometimes you have to wait till you come up and take the tube apart.
Get in there.
I'm watching this and just, I hope that's not permanent damage.
Well, it is for this. They'll re-terminate it, they said. They can cut it off and put it back on.
Talking to the masters.
I know, they had an engine fix in like 20 minutes today, so.
Yeah.
That goes the end of the section for the day. Whoop, whoop. And I think so. There you go.
There it is.
It
Well
is.
done.
Thank you.
Starboard inner. Was really impressive.
If you guys want to tuck it in the bio box, that'd probably be okay.
Okay, good. It's been gotten.
Yes, it's in the box.
Okay, lovely. My kudos to the pilots yet again for a
Right.
remarkable collection job. Thank you.
Mm-hmm.
And they will send Chris the bill for the new suction hose.
That's right. Be on the lookout.
Right.
Sure.
Well, that's definitely something you don't see every day.
Yeah. So we have about 40 minutes left. I say we make it to the top of this mound.
Yeah,
Make
I
that
would
the
like to
priority.
see that. Yeah, move
So-
kind of fast
Thank
until
you.
we get up
I'll
there.
talk to you guys
Yeah.
later.
Yeah, no
Thanks,
worries. No worries, Chris. Thanks for calling in.
Sorry about the tube.
Oh, jeez. They'll fix it. It's no big deal. What's up, John?
Alan and Stephanie,
Yeah.
I would suggest going to the top and
Yeah.
spending the last 45 minutes up there.
Yeah.
It'd be quite different,
Yeah.
or it might be the same.
Yeah, it might be. That's what I said. We're going to make our way up to. We're going to shoot for the top of this mound now, and then we'll look around up there before we leave. I'd like to get up there before we end the dive. We have about 45 minutes left. There you go.
What's your impression of the sponges that we've seen today? You know, a lot of different kinds, but most of them pretty small, or not so much?
We had a few large macro sponges. Let's see, we have a couple Ficalias. And let me look. Pretty diverse. A lot of them I don't know their names, so I don't know how helpful that is.
Some
But
Phoreas, maybe Euretid, definitely that Bullasoman.
the one we collected, the
Yes,
big, yeah.
I feel like we've seen quite a lot of small, like when we've zoomed in on the rocks, seen
Oh,
quite
yeah,
a lot
definitely.
of small, different,
Unidentifiables.
definitely unidentifiable from. I'm gonna save that. And then go take a look at the top.
All right, so we're going to store our collection skid.
Such a landscape.
Yeah, it's a rock boulder field, pile of rocks.
Yep, and now we will be making it up probably about another 50 meters to get to the top of this thing and spend the remainder of the dive up there, checking up the top.
Yeah, and I say we kind of just don't really look at anything too closely and let us at something new that we haven't seen yet.
You
maybe
mean
some snap
between
zooms.
here and there?
Yeah. Like between just some snap zooms, maybe. Like there's a couple of yellow sponges.
Yeah. It's hard to. Yeah, actually, if the snap zoom is possible.
Yeah. Okay. You want to snap zoom that yellow sponge?
Yeah.
Can we get a good zoom on that yellow sponge on your way past? One dead center. They should be the same species. Looks like a Liopathes maybe too. Oh, that's pretty.
Oh, that's nice.
A yellow hexactinellid.
Yeah, I think that might be Hertwiegia. It's sort of wide open.
Yeah, I think it's Hertwiegia.
Yeah?
Chris is back, so you let us know.
Okay. That's pretty cool. There's a worm inside.
Yeah,
a couple
really
of worms
nice.
inside.
So that's the first time we've actually seen that, even though we'd seen some other larger yellow sponges that made us think that we might be looking at Hertwiegia. When we zoomed in, it wasn't. But so this is, most glass sponges are nice, bright white colors, and this one is it's really distinctive yellow.
Okay, we're good whenever you guys are.
Thank you for that zoom.
Christina agrees. Hertwiegia.
Nice. It looks like this pile of rocks is a little more broken up here. They're not quite as large. More of like a 1 meter size, half meter size boulders. Again, we got Stichopathes. Sea Whip at like 8 o'clock. Maybe another Anipetherian at 6.
What is the minimum depth of this?
1331, John. Sorry, Nav, go ahead. Okay. Yeah. That's what we're shooting
Sounds
for.
great.
I'm going to get up there before the dive's over. We need our last water sample at the top there, too. Yeah, it's the same similar species we were seeing a little deeper. These yellow Stichopathes look like the most common. And that Paramuricid Gorgonian here, there's 3 at 9 o'clock. Each with their own ophiuroid in it. There's another Bathypathes at about 8, that feather-like one. And some more Liopathes.
The geology of this is really weird. We have to get a geologist to tell us what's going on. The multi-beam, just shows a flat plateau. You have this mound, maybe a half mile away, another mound that are perfectly conical, very typical coral mounds, and clearly this is not a coral mound. How did this thing
get here? I have no idea. Before you speed off at 9 o'clock, I've got another rock pen. It just went off screen. It is coming into screen right now. We just get a good video of that. So this looks similar to the one we got earlier, which is this rare morph that grows on rocks.
Yeah, just a bit larger.
Look, a purple gorgonian behind it, too. That's neat. Sweet. I would guess the same species, but it looks very similar.
It does look very similar.
Okay. Okay, we're good, thanks. There's some cool purple gorgonian behind it. Another black coral. See the black stalk?
Yep.
With the red flesh. It's probably, oh, that's a Stichopathes. There's maybe a stalk crinoid.
Yeah, still got a ways to go. Creeping up to the top of this deep mound.
Mm-hmm. It's like a random pile of rocks in the middle of the ocean. Let's see, 9 o'clock, I see a possible corallimorph right at the edge of the screen. Get a snap zoom on that. Nope, down further. Yeah, that's like an orange. It may not be corallimorph. It might be a, yeah, corallimorph. You can see the foot around the outside.
Foot.
Mm-hmm. Cool. Thank you.
Really large, about 10 centimeters across.
Yeah.
So now Scott thinks that was maybe a hormathid anemone.
Oh, hormathid, yeah. Isn't hormathia the fly trap?
Yeah, the flytrap is in that family, so I
Okay.
mean, I don't know for sure that it's in that family, but I do believe that is a true sea anemone actiniaria area and not a corallimorph and something close to that.
Thank you, Scott.
So Kimberly Galvez chimed in about the geology of this place.
Mm-hmm.
Well, that's right. Since we're on the edge of the Blake Plateau, this looks to be the exposed portions of the outcrop from the edge of the plateau. Could be broken up chunks of limestone that have since been encrusted with ferromanganese.
Mm-hmm.
Thanks, Kimberly.
These Liopathes, these orange black corals in the front of the screen.
As far as I know, Leopathes are the so far oldest known corals that have been aged using geochemical techniques. More than 4,000 years old, some colonies south of Hawaii.
4,000 years. That's incredible.
Mm-hmm.
So, I think still now the record is deep sea glass sponge. It was dated to 11,000
What?
years in age. Yeah.
Oh my goodness.
Wow, Alan, I did not know that, or I wouldn't have said that. So how do they age it?
I think that was isotopes on, because it's one of those ones with a giant single axonal spicule that's many meters long, and then it's laid down in layers, and I think the original layers were dated.
Looks like a bamboo coral here. Ooh, there's a cool sea star. Can you guys swing to the right behind the coral there? There's a poofy sea star at noon. He looks different than the other ones we were seeing. Is that one of those cushion stars? That's kind of squishy. Neat.
Wow. Looks
awesome.
Looks very soft.
Mm-hmm. Like that pillow foam in your sofa. Squish it. Hey, Chris, how are you? What do you got?
Hey, so that is a pterasterid. It's probably a pteraster, I think. So these are slime stars. Mm-hmm. If I collected one of those, or if you guys had collected one of those, chances are I haven't actually tested this yet with the deep sea ones in the Atlantic. But if we pick those up, they emit mucus as a defense mechanism.
Oh,
and
yeah.
So that would be fun, I think, for you guys if that was one of those things that we were going to test. You would probably get a box full of slime to work with when they come up. Or not.
It's not.
That's the fun part of testing these things in the deep sea. But I remember both pteraster and master can create a fair amount of slime when they're being harassed.
Are they
the animals themselves?
Go ahead.
I'm sorry?
I was going to ask if the animals actually
they're
squishy.
really weird on top of... Yeah, they are squishy, but they're also kind of crunchy because they have spinelets in the... they have what's called a supradorsal membrane, which is kind of a soft tent over their actual body surface, and it's held up by sort of calcareous, spiny tufts. And so basically, even though they're very fleshy, they still have all of these sharp, small bits in them, and so handling them is, I mean, like with pteraster, it's not such a big deal, but with hymenaster, it's a challenge because hymenaster is almost completely gelatinous. So that one in particular probably still had some skeleton to it. But like I said, their architecture, the body shape, is really strange because like they have they have a false layer over the top below, and then the actual surface of the animal is below this circus tent layer, which is on top. So they're very unusual, and there's a whole bunch of stuff associated with them that is interesting. But
awesome.
I'll let you get back to it. It looks like you have another rock pen.
Rock pen, and another Antipatharean in front of it.
So that's like another.
Have you seen this
like another interesting bunch of stuff going on. So I'll talk to you guys later. Hopefully, another starfish will come up before the end.
Okay.
Thanks.
We have not seen this black coral yet. This looks like Tenosidopathes, sort of, that we have on the in the shallower mesophotic area, but the polyps are way different. but they have that same growth pattern where it looks almost like you ripped a piece of a pine tree, like a Christmas tree growth.
Yeah.
Like a pine tree branch, but I don't know which one this is besides Antipatheria. That's as good as I can get it.
Well, this is a great view of it.
And then another rock pen.
Yeah. Calibelemnon.
Yep. Hopefully.
Calibelemnon franci.
Franci.
Is that what is on there?
This not?
Oh,
Oh, I don't know. It looks like something overgrown, like on the main stalk there.
Mm-hmm.
Maybe a parasite. It's overgrown a parasite or something. It looks like and a Bathopathes behind it. I saw Bathopathes too, right?
Mm-hmm. Is Batho or Bathy?
It's Bathy.
Yeah, Bathopathes.
Mm-hmm. Again, these rocks look like they're getting smaller as we get shallower.
Mm-hmm. We're up about 1,367 meters.
Mm-hmm. We have about, 15, 20 minutes left?
Got another yellow Paramaricia.
Mm-hmm. Looks like white sponge up here, maybe. And what was the name of this one with the stalk? Do you remember?
Oh, that's a Metallogorgia.
Metallogorgia. We've got a white. Look at this white sponge back there. Can we get a snap zoom on that weird sponge at center screen? Looks like a funky mushroom.
That is really cool.
What is that? That's weird.
No, I think that's
Hexactinellid?
I do think that's another hexactinellid.
Yeah, it looks like it's dying though. It's got that burlap texture to it. I don't have a clue what that is.
It's not conolasma, is it? Oh no, it looks like a tall notostrella, says Chris.
When you go wide on this, can you look under the rock to the right after you finish with the sponge?
Under the rock to the right.
After you finish with the sponge, you'll have to pull the camera back out. It's much closer to where we are.
So go full wide.
Reddish.
And then there's something reddish.
I'm wondering if it'd be Conorite. I don't know.
The single stalk here at 6 o'clock, or the one on top of
Below that black, yeah.
the single stalk? Can we look at that single stalk?
It might not be important after all.
Okay.
Well, they're all important.
Antipatherian, maybe? Yeah, Antipatherian of some kind. Looks like maybe a cut-off Stichopathes, but its polyps are huge. So, yeah, we're not sure what that is. I don't know.
It's an odd one.
Yeah. Okay, that's good. Thanks, guys.
Yeah, I agree, Stichopathes. But that's not at all what I thought. I was kind of looking for an Acanella bamboo, not that. Very cool. Thanks.
There's another Paramyrciid, maybe. It's at 9. And then we have 3 Stichopathes or whip corals right in the front. And we only have about 15 minutes left. Ooh, another Faria, possibly, over here. At 3. And then this one I cannot remember the name of.
Metallogorgia.
Thank you.
We're going to have to... So we got about 30 meters of relief to go. and I think about 15 minutes.
Mm-hmm.
I think that's another Hertwigia.
That was a big fish. Got a big fish at dead center. That looks similar to the one we saw earlier, but this one's skin doesn't look as... Oh, no, it is different, isn't it?
It's different, yeah. I think, anyway. Last one was more eelish, right?
Mm-hmm. This one looks more
This one looks
morid.
to me like a hake,
Hake
or something.
Yeah.
Again, I'm going way out on a limb. I'm not good on the fish.
One of these deep hakes.
Cool. There's a little piece of faria here at 8. Or 10, sorry.
Keep moving up.
This could be a... Let's see. Could this be a... No.
I don't have a clue. What do we think?
No, not that. No, it's not that. Because the tail's all wrong. Trying to look through pictures. Not the smartest way to do it, but...
He's a giant eye. And a big nostril, too. You can see the lateral line above the pectoral fin.
Mm-hmm.
That's how they sense changes in water pressure next to them.
Mike Vecchione agrees that looks like a blue hake.
Nice. Oop, and then a little guy next to him.
Oh, good.
That one looks more Lemonema-ish. It's a morid codling. Hey, somebody chime in?
Hey, this is Mike Vecchione.
Hey,
here.
Mike.
Can somebody chime in?
Yeah, Mike Vecchione here.
Go right ahead.
Yeah, that's the Antimora rostrata, the blue hake.
Blue hake.
Really cool.
Thank you.
Yeah, that's the Antimora rostrata, the blue hake. They're really common farther north at this depth.
Awesome.
They're really common farther north at this depth.
Can we get a quick zoom on the Allegorgia as we go by?
Like,
It's like it still has lower branches.
yeah, can we zoom in on this Gorgonia on that center screen? I think it's growing off that one. I think that bottom one is dead. Yeah, and it's growing off this. And again, we've got this little pink hermit. 2 of them.
Very
But
cool.
looks like it's growing off that.
I've never seen a Metallogorgia growing off another one like this. Awesome. And there's those dang hermit crabs again. All right. Moving on up. Thanks.
Yep.
Wow, that really is.
I wonder if is the other one a Metallogorgia too? Or is it a different species? It seems to be similar, but it's not quite the same growth. See the netting of the main branches looks different on the dead one.
I think it is, Stephanie.
You think it's the same?
It's got hydroid growth all over it.
Okay.
It's definitely a Chrysogorgiidae, which is the family that Metallogorgia belongs to, along with all the Chrysogorgia. But that's what it looked like. You had the 2 main bifurcation point branch.
Awesome. Thank you.
I'm really curious about all these hermit crabs, though. I said at the beginning of the day, I've never seen one crawling up on these tall axes like this, and now we're seeing all kinds of things.
I wonder if it's seasonal. Like maybe they're coming up, like it's mating season or something. And I don't know. Just wild guesses here. That's all I got today.
Special mystery mound hermit crab.
Mm-hmm.
And we still have we heard from anybody about what created this mound or what the idea is there?
Kim's had a commentary about it. She was talking about it being a buildup of rocks that were like left over from the. I don't know. I have to find her thing. Kim. Let's see. See if I can go find her comment.
Chunks of limestone blocks that could be the result of shedding from the outcrop and build up over time.
But why the mound?
Well, yeah, well, why the mound exactly?
Ooh, that's a big stick of pathies. Oh, we got a cool fish. Went down at 6. Little pink guy. He's in your list.
Yeah, I'm sure he is.
That is a Chonax.
Chonax, yes.
He looks like a balloon. He is a very grumpy fish. He looks very angry.
Oh, or is this a goosefish?
Yeah, John, what's up?
If you look at your multi-beam, you've got a long way to go to the top.
Yeah.
If there is hard coral, it's going to be on the tippy top, not this rope.
This is a gaper.
Gaper.
Chonacops roseus, it looks like.
Yeah. Go ahead. Don't think we're going to make it? Okay. 100 meters. Okay. So let's pick up, speed up, go as fast as you can. And we'll get as far shallow as we can get. No more looking. We're kind of going as fast as we can.
Understood.
No looking. Just go. Close your eyes. Yeah. Everybody close your eyes. We'll be at the top in a minute. So Chonax, is that what you had? Is that was that right?
I thought it was Chonacops, but that's what
you can get me the name later. That's fine. Pink.
Yeah.
Yeah.
The rosy coffin fish.
I figured. I just timed it bad. We just need to barrel through here. Oh, that we may not make it to the top. So we're going as fast as we can. Yeah, we've been moving nonstop since for a while now. So we're just going as fast as possible. And we were trying to get to the top of this, but I don't think we're gonna make it. We only have about 8 minutes until we have to come up. Here's a cool yellow one. Yeah, I'm not allowed to look at it though. Gotta go quick. Don't look. Yeah. Yeah. That looks like Leiopathes to me. There's a little yellow Faria possibly. Some white Faria here at 6.
Mm-hmm. Here's where we're going to start seeing the Medusae.
Mm-hmm. So you can collect more?
Can we?
No. Yeah, the boxes are full, and it's like, yeah. So I can see up this hill, and I see some gorgonians, but I don't see any. If there was Lophelia, it'd be white. You'd be able to see it from here far away. Ooh, here's a tube, a white tube, hexactinellid.
Oh, so another euplectellid, maybe?
Oh, that's funky looking.
Oh, no, it's not. Doesn't have a sieve plate on top. Just a thin...
Just a tube. Euplectellid, and there's another one too, isn't there? Heterotella? The wedding sponge.
Oh, euplectella and
regadrella.
But yes, but it's not one of those because it didn't have a...
He asked.
Oh, sorry. I'm sorry. I was a little busy in the chat room. Sorry.
That is a fish. A little scorpion fish, maybe? No, it's one, I think. No, I have no. It's some kind of hake or... Oh, neat. Look at his dorsal fin is moving around. Look how long he's got these tendrils, too, underneath.
Mm-hmm.
The feelers, maybe?
It's a really interesting looking fish.
Mm-hmm. He's also got chew on his nose coming out on his face. 2 feelers.
Hmm.
Come on, get out from under that rock. I want to watch you swim. And you just swing a little bit to the back. You just passed over a coral. I wanted to get a quick look at that guy, that white guy right there. Okay, Gorgonian.
Look on the bottom. There's a bunch of rubble.
Bunch of rubble on the...
Try to zoom
Yeah.
in on that.
That looks like it could be Lophelia rubble here that it's attached to.
Corallium again.
Yeah.
There's some sort of rubble all over the...
Yeah, there is. Can we get zooms on this rubble here? That might be coral rubble. Yeah, it is hard to tell. Let's see. I'm not sure what it is. It could be a coral rubble, John. I don't... Yeah, right
There
here.
in the lower right.
Yeah.
Actually,
I don't think
Go
it's
to the top.
Lophelia, though. Yeah, okay, guys, okay. Stop looking, we gotta go.
Science, if you look at stream 3 or look at the Sirius view, you'll see that Sirius hasn't stopped. Sirius is still moving.
Mm-hmm.
So we're not stopping.
Okay.
We'll just remember to...
We're trying to get as shallow as possible, and then right before we come up, we want to snap that last water sample. Some different black corals, it looks like. It looks like we're losing our rocks, too. Getting more sedimenty.
Yeah, it's interesting. That
There might be a little white coral there. Mm-hmm. Yeah, a little Gorgonian. Nope. Snail.
nice little gastropod peeking in there. Great.
Okay, can you
just snap the water?
Yep, give us a water sample. Thank you.
Thank you very much.
Either way, it doesn't matter.
Yeah, this is fine.
Mm-hmm. Dive 5, sample 0. That should be 9. 9W. Got it. Thank you.
Thank you very much. Logged. Whoops. Got the error. Logged. Dive concluded. Well, that was awesome.
Yeah, it was. So sorry, John, we didn't make it all the way to the top. It was totally my fault. We were taking too long.
There was a lot to see.
So just to recap, if you're just joining us at the end of our dive today, this is actually our second dive today because our first dive, we had a mechanical failure on the ROV, so we had to recover it and then relaunch in the same spot. Let's see. We are 174 miles from Jacksonville along the Blake Plateau, along the
Eastern edge?
Yeah, the eastern slope. And we traveled from about 1432 meters at the base of this mound up to, we got up to 1350?
Yeah.
We took 9 samples. 4 of them are water. Right?
Yes.
Let's see, the total rise for this mound was about 101 meters. We didn't quite make it to the top, though. 18-degree slope. The length of this feature is 620 meters. And then we had planned a 545-meter dive, but I'm not exactly sure how far we actually made it. That would be post-dive data. I'd have to find out.
And we were curious if this was going to be a biogenic mound, but it turns out it was not. It was very rocky.
There may have been some coral rubble there at the end, so there might be some coral at the top of it. And unfortunately, I'm a slacker, and we didn't get it there.
We had a lot to look at.
Yeah. We are going to have a post-dive call and a pre-dive for tomorrow call after this. I believe as soon as we're done broadcasting. So if you want to be involved in tomorrow's dive, go ahead and stay on the line or call in. We are going to one of the deep dives on the Blake Spur tomorrow, and we'll be on the Blake Spur for the next couple of days, and then we'll make our way back across the Blake Plateau. So go ahead and stay on the line. We'll be with you guys shortly while they do recovery. And I think we're going to go eat dinner as quickly as possible before we have to process all these samples. Okay, so thanks for diving with us today. It was really fun. We hope you join us again tomorrow. Anything else, Alan?
No, not a whole lot. Just thanks a lot.
Yeah, thanks, and thanks for chiming in and being involved and helping expand.
Tomorrow we're going below 4,000 meters,
yes,
so it's going to be
one of our deepers.
More exciting.
Yeah, one of our deeper dives. So just hang on the line. We'll be with you shortly.